touch in the past. And today, I'm going to be talking a little bit more about the tools we use to study these. So when we took you guys into the lab, we showed you some of the big microscopes and rigs that we have for measuring the electrical activity of cells. This is a way of studying our touch and heat sensitive neurons, and it's also a way of studying the molecules that are involved in heat and touch. And so right now I'm going to talk a little bit about the science of how this works. It's a tool called electrophysiology, so electro for electricity, and physiology for body function. That's what people talk about when they talk about physiology. It's all about how the body works. And after this, we're going to be using electrophysiology to study touch responses in cockroach legs. So first, just a little bit of a review. So Willem told you on Monday that neurons talk to each other via these chemical connections called synapses. And when one neuron is activated, an action potential fires, and it releases neurotransmitters. These neurotransmitters cause charge to flow into this next cell. And if enough charge flows into that cell, then you can get a large electrical impulse called an action potential. This is synonymous to like nerves firing or something like that. This huge electrical change that happens with charge entering the cell at first and then eventually leaving the cell. This electrical signal causes the axon terminal here, the synaptic terminals, to release neurotransmitters, and that's how this neuron talks to the next neuron. This is what happens in the brain. It's a little bit, our sensory neurons are a little bit different. So, let's see. Rather than receiving input from another neuron via neurotransmitters, the way that it receives input is through a physical stimulus. So maybe through some mechanical stimulus or through a heat stimulus. This is how you can start to get positive charge entering the cell. And again, if you get enough positive charge entering the cell, you end up getting this large electrical impulse, this action potential. The nerve fires. And this nerve firing leads to, again, neurotransmitter release at the synapse, which can then tell another neuron to fire as well. So this is what we're dealing with in the sensory neurons. Rather than receiving input from another neuron, we're receiving input from the environment in the form of a physical stimulus, either temperature, hot, cold, different types of touch, pain, and stuff like that. Is that cool? So the way that we can measure, or that we can study this in a quantitative way is we can stick an electrode right outside of this neuron. The electrode picks up, what an electrode does is it can pick up the voltage between two given points. And this voltage changes depending on the flow of charge into the cell and outside of the cell. So when you get your physical stimulus, maybe like putting your hands near a fire, or hopefully not somebody hitting your thumb with a hammer or something like that, you get these nerves firing, and the electrode can pick up on this firing. And using this, we can study the rate of firing to get an idea of how we're sensing touch or how we're sensing heat. Now, so this is a picture of what's going on, but the way that the data looks is a little bit different. So this is what the data looks like. Our bodies run on electricity, so if we stick some electrodes in the body to see what electrical activity is going on, we have some baseline electrical activity going on. Can some people think of parts of the body that really rely on electricity? The heart. Yeah, the heart is a great example. So you have this intrinsic pacemaker activity that helps the heart to pump synchronously, different parts of it to pump in time. our nerves definitely rely on electricity, and our muscles too. So similar, the heart muscles rely on it, but our muscles outside of our body rely on it too. So there's a lot of background electrical activity in our body and also in the environment. But what we're looking for here in these extracellular recordings are large changes in voltage that are related to nerves firing, these action potentials firing. And what we can get from this is the spiking frequency. So how often these nerves are firing per second. This can tell us something about how a particular sense is touched, how different parts of the body sense touch, and then also how they adapt to this stimulus over time. So we talked a little bit about adaptation in previous lectures. Looking at how this spike frequency changes over time is a way of looking at adaptation too. So what you guys are going to get to do this right now, and to do this we're going to be studying touch sensation in a cockroach leg. And so Willem is going to do the dissection in the back. If you feel comfortable looking at this cockroach dissection, definitely go up front and check it out. And if not, that's cool. But what Willem's going to be doing is he's going to be anesthetizing this cockroach with cold water. And he's going to be cutting off this leg. And then we're going to be using this leg to study touch and adaptation to touch. So I think Willem is ready for you now. You guys can stand up and go to the back.